everyone to one more week of the Behavior, Evolution, and Culture Speaker Series. We're almost done with the year. We have 2 more talks. So before I introduce today's speaker, I will give you a preview of what's coming next week. Also, there is a sign-in sheet going around. Please, if you haven't already, sign it. And let's see. I guess you don't need to look at the website, but soon there will be a list of talks for next year, which you can find at our website, which is bec.ucla.edu. And next week, June 3rd, will be our last talk of the year.
No. Next week's a holiday.
Oh, sorry. Yes, sorry. Next week is a holiday. Don't come next week. Sorry, June 3rd, 2 weeks from now, is our last talk of the year. And we have a tradition in the department that when we have students who are finishing their PhD, that we don't have formal defenses here, but we have students who give a BEC talk in lieu of a formal dissertation defense. And so our very own Katrina Kayakaite will be giving a BEC talk, and we have a title for it, Testing Hypotheses About Social Cognition with Observational Data, Coalitions in White-Faced Capuchin Monkeys. So look forward to that in 2 weeks. Otherwise, I think that's it for the announcements, and I'm very pleased to introduce Courtney Meehan, who's here from Washington State University, and she's going to be talking to us about the social worlds of infants, moms, and microbes.
Well, thank you all for coming and having me today, and thank you, Brooke, for the invitation, and Sonia for helping organize this. As Brooke just said, I'll be talking about the social worlds of moms, infants, and microbes today, and this is pretty much a mixture of all the different legs of my research. And so, to give you a bit of background, my overarching interests have centered on evolutionary cross-cultural explorations into the role of early environments and their relationship to maternal life history strategies and infant and child development. Generally, this has been an interest in human cooperative breeding and how early and social environments are associated with maternal energy expenditure, infant or child growth, social emotional development, and more recently, how they have been associated with our microbiomes. I've actually been working on this longer than it looks like because it was pretty much going back for a second PhD to figure all of this out. But over the last now approximately 8 years, my work has really centered on how we can understand human microbiomes and their relationship with early development and early environments. And so, as an anthropologist, I believe in the essential nature of cross-cultural work and long-term field work. As such, I've worked with the Aka hunter-gatherers in the Central African Republic for almost a couple of decades now. The times are changing for all hunter-gatherers, but many of the Aka remain mobile, moving camp several times a year, but certainly the Aka have experienced some cultural change over the last several years. The Aka reside in the Congo Basin rainforest, and on the periphery of that rainforest are groups of horticulturalists who I also have worked with for the same amount of time. In recent years, in part due to violence that erupted in the Central African Republic, I shifted to work in Ethiopia and started a project both in the highlands of Ethiopia in a rural community of agropastoralists and also in the urban area in the lowlands. And I've also been working in the US with a Washington and Idaho population, and over the last 5 or 6 years I've been co-directing a large interdisciplinary and international study on human milk composition and maternal-infant microbiomes called the INSPIRE Project, and I'll talk more about that in a bit. And so with that background done, I'd like to talk about some of the work that we've been doing related to human milk components and really the social worlds of moms, infants, and microbes. It's only been a few years since the vast majority of scientists have recognized that human milk contains bacteria. It was long thought to be sterile. In the first paper identifying bacteria in milk using modern methods are actually, it was from my colleagues, Shelley and Mark McGuire, Janet Williams, and it was led by Mark's student, Catherine Hunt. And so this was published in 2011, and so in the years since, there's been an explosion of research, first trying to figure out whether or not milk actually does contain bacteria, and second, what are the factors influencing variation in those microbial communities. And so we now know from that paper and subsequent papers that milk does contain a diverse microbial community. Yet we still know relatively little about how the human milk microbiome, or HMM, is shaped, and even less about its role in infant health or in mammary health. And so, to give you a bit of background here, multiple origins of the HMM have been posited. This is a figure from Fernandez et al. And origins may include the maternal gastrointestinal tract, skin microbiome, or the infant oral microbiome, which may occur via flow back into the mammary gland during breastfeeding. It's likely, however, that all of these are sources of the milk microbiome, and potentially others that we haven't yet identified. But the increasing attention over the last several years on the human milk microbiome has really resulted in an expanding literature focused on the factors associated with the HMM. We now have fairly solid evidence that geographic location, diet, whether or not the mom is obese or not, the gestational age of the infant, and whether or not the mother used antibiotics all affect the diversity and community composition of the human milk microbiome. Studies on birth mode and time postpartum have shown inconsistent results in the literature. It might be due to methodological differences. We definitely need more longitudinal studies to be able to tease that apart, but those haven't quite been determined yet, but I'll talk about what we found in our study coming up. Oops. And so oops, sorry, second. I feel like I've just lost a slide there, but what I'm really interested in is the anthropology of milk, that intersection of human biology, ecology, sociality, and behavior. Human sociality and behavior, to a large extent, have yet to be fully considered in regards to human milk composition, and particularly in regards to the HMM. Yet, I view it as incredibly important that we investigate this, as milk serves as one of the earliest and most continual sources of microbes to the infant during infancy. And milk may be priming the infant for the environment in which they will be reared, which includes their social environment. That social environment is critical. We are successful as a species due to our ability to cooperate and live in social groups. Humans are, in fact, a cooperative breeding species, simply meaning that we rely on the assistance of others to rear our offspring to independence throughout our evolutionary history and in many parts of the world today. But so essential is that cooperation in child rearing that Herdes argued that cooperation was likely a pre-existing condition to the evolution of altriciality in our infants and to the evolution of extended child development. Our social worlds and the way in which we rear our offspring were likely in place prior to our current development patterns, and thus were also likely to predate the evolution of what we think of as normal human milk factors and normal human milk composition. As an evolutionary anthropologist and behavioral ecologist, I'm interested in the intersection of lactation, milk components, and the environment and culture. Human milk has evolved for millions of years to support our unique needs of our infants, but that evolution has not occurred in a vacuum. It's occurred within the social environment in which we've been rearing our children. Although not focused specifically on the HMM, how human life history characteristics and patterns affect milk composition has been gaining ground in recent years among biological anthropologists, thanks to some really amazing work done by my fellow milk-centered anthropologists. And so overall, it's a really exciting time to be doing this work. And in terms of the microbiome, the idea that our social worlds are important isn't coming out of nowhere. although it hasn't yet been applied to milk until our group started working on it, we've already seen that social environments are important to the shaping of our gastrointestinal microbiomes, which is actually really our fecal microbiomes. But the majority of literature has focused on animal studies, but we've seen among baboons, chimpanzees, lemurs, bees, ants, and some studies among humans that social membership is associated with GIM or IFM community composition, and that social interactions are correlated with intra-individual microbial similarity. In terms of the human studies, we've seen quite a bit of variation in the literature. We can start with siblings here, as they tend to be the individuals that people have focused on most often. Larson and colleagues showed that the number of siblings was positively associated with GIM richness and diversity. However, Megan Azad's group showed that sibling presence was negatively correlated with GIM richness and diversity. My graduate student, Avery Lane, working with our large international sample, just published a paper last week that showed no relationship between siblings and the diversity of the infant fecal microbiome, but did note several taxa that are associated with it. Family size has been shown or observed to have no relationship with household member GIM diversity in a study by Song, but studies centered on community composition have found that the number of family members are positively associated with the relative abundance of bifidobacterium. Increased lactobacillus, bifidobacterium, and Bacteroides, with a reduction of Clostridium colonization in the infant GIM. So differences in diversity and community composition are again perhaps due to methodological differences, or perhaps they're simply differences in the environments in which these children are being reared. For example, in some populations, children are going to receive maybe more sibling care. In other populations, older siblings are off at school and not interacting with their younger siblings as much. And so we have quite a bit to do to be able to tease this apart. But I've always been interested in these larger social groupings, kind of the community as a whole. And so Schnorr and colleagues looked at or touched on this in a paper in 2014, where they found that Hadza had higher intergroup similarities than an Italian cohort, which at the time the authors attributed to close social and cohabitation ties common in hunter-gatherer populations. And so with that in mind, but before we return to our discussion on milk, I want to take a detour away from milk and spend a bit of time talking about what we know about the social and behavioral worlds in which we reared our children throughout our evolutionary history, and why they may be of particular importance to shaping milk factors, and in particular the human milk microbiome. I'm going to focus on just 4 key ancestral human caregiving traits that we assume were essential to our success as a species, and those include continual contact, cooperative breeding, on-demand breastfeeding, and co-sleeping, all of which influence infant microbial exposure and possibly the composition of human milk. And so to do so, I'm going to rely on kind of my life as an anthropologist more, and talk about the work that we've been doing over the last decade, trying to document in really fine detail what infancy looks like in different cultures around the world. And so we've been doing this among the Aka in the Central African Republic, and among the Ngandu, also up in the highlands of Ethiopia, among the Sadama agropastoralists, and in the United States, in the Moscow-Pullman region of Washington and Idaho. So if any of you haven't come up to the Palouse, it's actually a beautiful time of the year. It's that bottom one where all the wheat fields turn green, but it's a pretty rural community. We have 2 major universities dropped within 7 miles of each other here in the middle of the wheat fields. Combined, the population's probably about 60,000, about 25 in Moscow where the University of Idaho is, and close to 35 in Pullman, but fairly rural communities in the US. And so since I've started doing anthropological research, I've spent quite a bit of time focused on doing naturalistic behavioral observations of infants and their caregivers. For all of these studies that I'll be talking about, we have done naturalistic focal follows. We record infant behavior and their interactions with their caregivers every 30 seconds. We follow infants throughout the day. It's actually spread over several days in timed units. But if they go hunting with their mom, we're hunting with their mom. If they're in the fields with their mom, we're there too. In the US, if they happen to go to a party, we go to the party. So there are required breaks and observation segments that are purposely spread over multiple days, but we cover 9 out of the 12 hours of daylight, at least daylight for those populations near the equator. And so over the last several years, we've enrolled 263 infants in this study, which results in well over 250,000 observations on infants and their interactions with their caregivers. We recently just finished entering one of the final field seasons and the US data, and so much of this is unpublished, but we're working on it. And so the data that I'll be talking about over the next couple of slides, most of it, but not all of it, comes from these behavioral observations. And so to start, this is actually a figure from a chapter that I wrote with Barry Hewlett and Ed Hagen a couple years ago, where we were looking at whether or not ACA caregiving patterns had changed over the last couple of years, over the last couple of decades, I should say, given the changes that they've experienced, either politically, socially, and culturally. We found that there hasn't been much change, but regardless, we can see that the ACA on your left here are in physical contact with their infants about 90% of the day. If we look to other hunter-gatherers all the way to your right, we see that the Baka are in contact with their infants about 80% of the day. Horticultural populations tend to be around 60% of the day. And then down here, were the Euro-Americans, around 30% of the day. I do want to be cautious for a second, and I'll talk about that 30%, because our recent analysis from our US data puts it probably more around 60%. And the difference between those two, or at least my hypothesis for the difference, is that our US, Washington, Idaho population versus that Baltimore population, we've enrolled moms in a very intensive, or they volunteered, I should say, in a very intensive milk composition study that lasted several days. I think we're self-selecting for moms who are excited and interested in human milk studies, probably practicing more intensive or maybe attachment parenting styles. And so I think some of this is a sampling selection, why we're up there around 60, and previous studies have shown it much lower. With that said, I think that our moms up there around 60 are probably more comparable to our other populations, but perhaps less comparable to an overall US population in terms of behavior, I mean. And so you might also notice that there aren't very many populations on this list. And for those of you that study caregiving, you're probably thinking about a few that we could have put up there. Unfortunately, anthropologists, we tend not to collect comparable data. And so it's incredibly difficult to dive into the literature and find data to compare to each other. And it's one of the reasons I've spent the last decade kind of painstakingly working to do this across different cultures so that we can compare data collected by the same methodology to try to understand what things look like cross-culturally. But, so that was physical contact. Now switching gears, but related to look at cooperative breeding. Cross-culturally, humans are born into and engage with and receive care from multiple individuals. In small-scale societies in particular, the mother-infant dyad is often entrenched in really deep social networks comprised of individuals who occupy different genders, caregiving categories, familial or non-familial roles, but the degree and the breadth of these networks obviously varies cross-culturally. Here you can see the number of individuals who were in physical contact with infants across daylight hours. So the y-axis is the mean number of individuals who came into physical contact with infants across daylight hours. The numbers on the bottom represent age categories. So 1 is less than 6 months, 2 is 6 to 12, less than 12, 3, 12 to 24, 24 to 36, and 36 to 48. You'll see that there's variation when we get to the agropastoralists in the US in terms of the number of age categories is kind of bringing together a variety of different studies, and so not all studies focused on 0 to 4 years of age. But what you can see is that caregiving, particularly in our small scale societies, is highly distributed. We are not seeing just grandmas engaging with infants. We're not seeing just sisters or even just juvenile girls engaging with infants. What you can see is, for example, let's see if I can get this up, ACA infants in late infancy have about 5 juvenile girls engaging with them across the day, but juvenile girls are by far not the only type of caregiver who's engaging with them. And so this gets a little dense to look at, but I wanted to give you an idea of what it looks like across early development. But if we just take this and focus on our first 6 months of life, we can see that infants have large and diverse caregiving and social networks that are not dominated by one category, but we can see that there is variation. In the US, it's almost half. We only have around 3 caregivers per day in the United States, and that's almost half of what we see amongst the next lowest, which are the Sidama agropastoralists. And interestingly, the Sidama practice a period of postpartum isolation is the wrong word, but they stay home and they don't leave the house for a few months. It's important for moms and babies to rest in the home and not to be out and about. So they are fairly isolated, but even so, their infants are engaging and being touched by a variety of individuals across the day. It's certainly not at the level we see among the hunter-gatherers and horticulturalists, but it is distributed. I should also note that in the US, it's most heavily dominated by those who are residing in the household. And you only see, on average, kids have less than 1 caregiver per day that's outside the household. And I shouldn't say necessarily caregiver, but one who comes into physical contact with that infant. But diversity in the number of individuals who touch or engage in physical contact with an infant might not actually represent diversity in caregiving. So for example, we might see all of these individuals engaging with the infant, but perhaps moms are doing 90% of the caregiving, or perhaps allo mothers are doing 90% of the allo-maternal care. And so here what we have is, again, just our infants less than 6 months. And the top graph looks at the frequency of observation, simply the number of times we saw particular individuals in contact with their infant. And we see that moms dominate the frequency of contact with their infants less than 6 months. This is not surprising. They're all breastfeeding moms. The bottom looks at the mean proportion. So it's the proportion of total care. Again, moms are providing the vast majority of care that infants receive. So moms are providing 63 to 89% of the physical contact that infants get, which means that allo mothers are contributing 11 to 33%. 11% in the US sample and 33% in the horticultural sample, which is pretty much on par with the ACA as well. But I think it's important to explore the allo-maternal behavior just by itself, particularly before we get back into the microbiome conversation. The literature is ripe with studies, including some of my own, that show that allo mothers provide the mother-infant dyad with support, assistance, protection against environmental shifts. They improve child survivorship, not always, but frequently. And they frequently, again, but not always, improve the health of children. The care offered by these individuals indicates a direct pathway by which they might influence microbial exposure. And so if we explore the percent of allocare by caregiving, the bottom graph here, we can see that care is most distributed among the hunter-gatherers and horticulturalists and continues to become less distributed across other populations to the point of dads contributing 46% of the care, percent, not frequency, of the care in the U.S. With the U.S. Exception, however, not one allo mother really dominates these other populations. And so in small-scale populations, caregiving is not just distributed in terms of the diversity of caregivers, but it's also distributed in who's investing in those infants. And so if you're interested in cooperative breeding, and maybe not so much in the microbial aspect of this talk, I'm actually really excited about this because for many, many years, we've been talking about specific caregivers as being the essential caregiver. And what all of these data show, and again, this is some detailed observations, that what all of these data show is that caregiving in small-scale populations is distributed across caregiver categories and in terms of the investment. So we've done physical contact and cooperative breeding. I kind of already said that, but allomaternal caregiving is distributed across caregiving categories. But as households and moms become more isolated, allocare becomes more centered in the household. So we've done physical contact and cooperative breeding. I'd now like to talk about breastfeeding. Our breastfeeding habits have also markedly shifted in recent history. These figures represent the number of breastfeeding bouts or episodes across a 12-hour day. So every column here is an infant sorted from youngest to oldest from 0 to 6 months. The figure on the left is the ACA, and the figure on your right is the US population. And I only put those two up or it becomes impossible to see. But this is, and then from the top, the rows are the hours of observations. And so what these heat maps are showing is the darker the square for this particular heat map, the more breastfeeding bouts that occurred during that observation period. And so what we see is that the ACA hunter-gatherer infants have 2 times as many breastfeeding bouts or episodes across the day. And if we look to breastfeeding as a form of communication between mothers and infants and a way for infants to signal to their mother their microbial exposure, perhaps more frequent breastfeeding might enable infants to signal and communicate more efficiently or effectively across the day. We don't know that yet, but it's one of our hypotheses. And then lastly, co-sleeping. 70% of the world co-sleeps with their children, and that number goes up to 100% when we start to look at small-scale societies. Throughout our evolutionary history, co-sleeping provided thermoregulation and protection for infants, and it also enabled continual nighttime breastfeeding as well. These data are not from the behavioral observation data. These rather come from the international INSPIRE study that I'll talk about in a few minutes. But what we see here is that separation is rare until you move into urban areas. You start to see nighttime separation, even in some of our urban African populations between moms and babies. And then we really start to see it as we move into Europe and into the United States. And so basically the majority of mothers and infants bed share. Nighttime separation is really only occurring in Western and or urban areas. And so how is all of this related to milk? And so now I'd like to return to the conversation on the human milk microbiome, and I'll discuss 2 studies that center on how the environment, geography, and our social and behavioral worlds are associated with the HMM. The first one that I'm going to discuss we published last year, and it was focused on the hunter-gatherer milk microbiome and horticultural milk microbiome and the CAR. And the second one we're just finishing up, and so these data are unpublished right now too. But we argue that there is no normal human milk composition. For a long time we've assumed that milk composition, or there at least is some degree of normality across women, and we argue that there is no normal human milk composition, and particularly no normal human milk microbiome. That microbial communities are likely adapted to local ecologies. And when I use that term, I think very broadly about it, to our physical environments as well as our social environments, and even to behavioral variation. If milk is offering infants cues to their environments, and mothers and infants are able to communicate microbial exposure through breastfeeding, milk that is responsive to shifts in the environment may offer infants a fitness advantage. And so in this first study that we conducted in the Central African Republic among the Aka and their neighbors, we were interested in first understanding whether or not the human milk microbiome varied cross-culturally. When we collected these data, we didn't have any other studies to look at to see whether it varied across societies. And we collected these data in 2011. It took a while to get that out, but we were interested in how several factors might be associated with that variation as well, and in particular the diversity and the community composition of mother's milk. And so we looked at whether subsistence type, which in this case corresponds to variation in ethnicity, influenced the human milk microbiome, whether we saw seasonal variation. The milk samples were collected both in the dry and wet seasons, and so we were able to look at that. And since both populations reside in or on the periphery of the Congo Basin rainforest, but they have different childcare practices, we were also interested in whether there was variation in the human milk microbiome based on behavioral patterns. And so we collected milk samples from both populations, and we analyzed 41 mothers' samples and characterized their milk microbial communities. The first thing is the milk from CAR looked like milk, which was good. And it was similar to other studies in that, for example, strep and staph, those top two up here, dominated the microbial community. But it also differed from previous studies in Western populations. For example, we found a high relative abundance of rhizobium in the milk of Central African women, which had not been seen before. In our international study, we've also identified rhizobium, which is a soil bacterium, in the milk in a couple other populations. And so we are seeing that even though this popped up as one of those key ones that kind of surprised us, that we've been seeing it in other populations since. And so we first explored whether subsistence type, season were associated with variation in the HMM, because again, these populations interact with each other and share to some degree the same environment, but certainly the Aka are spending more time in the forest. What we found is that certain taxa, such as Gomella and Kokuria, are more abundant among the hunter-gatherers, while other taxa like Anhydrobacter and Lactobacillus are more abundant among the horticulturalists. We also found that Burkholderia is more abundant in the dry season than in the wet season, which we think is related to subsistence activities during that time. Whether or not this is related to genetics, environment, diet, or other activities, we simply don't know yet. We are really just at the beginning of these analyses. And as we move forward, we'll hopefully be able to tease this apart. But for the moment, we are simply trying to identify what exists in milk around the world and to try to understand some of the factors that might be associated with variation. But we are also interested in exploring how the infant's allo-maternal network and the frequency of care they receive from their mothers and others was associated with the HMM. And so we had several mother-infant pairs that were also participating in the behavioral studies. And so using the behavioral observation data on mother-infant dyads, we dove into how early social environments and the caregiving world was associated with HMM diversity and community composition. We centered our analysis on 9 dimensions of infant care, which we ended up reducing into 2 principal components. Briefly, Principal component 1, which explains over 50% of the variation, can be thought of as an infant's caregiving network and allomaternal caregiving environment. Principal component 2 appears to represent variation in care that comes from both mothers and others, but it is primarily being driven by maternal care and can be thought of as the intensity of the infant's caregiving environment. And so what we found is that the density and frequency of infants' caregiving experience was significantly and positively associated with HMM diversity. Specifically, we found that the milk of mothers with infants who had more dense allomaternal networks and infants who received more allomaternal care had greater HMM alpha diversity. In contrast, PC2, which again represented the intensity of the caregiving environment, was not associated with diversity or evenness, but did show non-significant trends for 3 of the 4 richness estimators with similar effect sizes as the other analysis, indicating a possible relationship between the intensity of an infant's caregiving environment and the richness of the mother's milk. I shouldn't say the richness of the mother's milk, I should say the richness of the HMM, sorry. And so we also found that the density of allomaternal networks and the frequency of care was associated with the composition or the community composition of mother's milk as well. For example, we found that Diella and Xanthanum bacteria are of greater abundance in the milk of mothers whose infants have more intense caregiving networks. And so with that in mind, I would like to shift gears, and we'll pull this together at the end, but we've also begun to explore these questions in the INSPIRE study. And the INSPIRE study is an international and interdisciplinary study that my colleagues and I direct that occurred in 4 continents, across 4 continents in 8 countries at 11 sites. We collected milk and infant fecal samples in rural and urban Ethiopia, rural Gambia and urban Gambia, Ghana, Kenya, Sweden, Spain, Peru, and in Washington and Idaho, and in California with a Hispanic population. We enrolled 412 mother-infant dyads in this study, and the analysis I'll be mostly talking about today focuses on 346 mother-infant dyads from 10 sites for which we were able to obtain all of the social and cultural and behavioral data for. In order to enroll in this study, moms and babies self-reported as healthy. Infants were between 1 to 5 months. No antibiotics could have been taken by the mother or the infant in the last 30 days. Moms had to be breastfeeding more than 5 times per day, and neither could have reported any illness within the past 7 days. So no fevers, no diarrhea for moms, no pain in the breast or hardening or red streaks on the breast. And so to do this study, we standardized methods as much as humanly possible. We created all of the kits at the University of Idaho. We put all the same gloves in the kits. We even, for the infant fecal samples, we even sent diapers all around the world. And we use Walmart diapers because they don't absorb anything. And it made it easy to collect those fecal samples. So everything was pre-made and arranged and then sent out to the sites so that we could limit the variation when it came to the collection. So in terms of the milk samples, moms were able to pump via sterile collection kits or manually express, but their hands were gloved, and that depended on where we were, on what site, and what was culturally appropriate there. All moms' breasts were pre-cleaned. We used sterile collection kits and cups. Everyone was gloved. We aliquoted into sterile tubes via sterile transfer pipettes. And we had great success at this, except for one of my sites, unfortunately. In rural Ethiopia, we lost electricity, and we ended up having to chemically preserve that milk. Kimberly Lackey, who is now a postdoc on this project. If you're interested in chemical preservation of human milk, she actually just published a paper on this a couple years ago, which highlights the chemical preservation method we used for the rural Ethiopia site. And then after all the samples, milk samples were frozen immediately following collection, and they were all shipped on dry ice to the University of Idaho. So in terms of the survey and demographic data, this project was a huge undertaking, and so we made sure to collect as much as we could on that one time because we knew that this would be hard to go back and redo. So we collected reproductive histories on all the moms, household composition, birth experience, child care practices, breastfeeding patterns. We have dietary data, 24-hour and 7-day recall. We have food insecurity data, information on health-seeking behaviors, infant and maternal health histories, and anthropometric data on moms and infants. And so our overarching question for this study was, how does milk composition and milk and infant fecal microbiomes vary globally, and what is driving that variation? And so far we have analyzed the milk for protein and lactose. We recently published our human milk oligosaccharide paper looking at variation in human milk oligosaccharides across the sites. We've also recently published the paper on the immune factors in the milk, again looking at variation across the sites in human immune factors. We are doing studies on antibodies to common infections that we find in milk, and we also collected genetic data on these women. So we're currently analyzing those data to try to get a better handle on variation in HMOs, but we can do this with any of the milk factors. And so that HMO paper is being led by my colleagues Brenda Murdoch and Janet Williams. And then just last month, very excited, we published the characterization of the HMM, the human milk microbiome, and infant fecal microbiomes on all the sites. And this paper was led by Kimberly Lackey. This was part of her dissertation. As I mentioned, she's now a postdoc on the project. In this paper, we not only characterized the HMM and IFM across the sites, but we also looked at their relationship to each other. And this slide right here is the HMM data. And not surprisingly, we found that there is geographic variation in HMM. And perhaps more importantly, we looked at the relationship between the two. And so in the top left, you can see the overall relationship, and then each of the other boxes is one particular site. But what we found is that there are strong positive associations between the complex communities. We found that there were limited associations between individual genera, but that in the milk and feces, but community level analysis suggests strong positive associations between the complex communities in these samples. But now that those characterization papers are out, we get to start to have a lot of fun. And so the paper I'll be describing to you now, we're just finishing up, and we are able to now dive into kind of those overarching questions that we were interested in. And so here, what I'm going to be talking about is some of the environmental, individual, household, and caregiving characteristics and their relationship to the HMM. Looks like the colors didn't quite come out in that. That's okay. So we controlled for a geographic region, whether moms are from Africa, Europe, or the Americas, and we also looked at maternal characteristics, age, parity, time postpartum, BMI, birth mode, whether they gave birth vaginally or by a cesarean. We looked at household features, how many household members were residing in the home, whether or not animals were present in the home. And then we looked at caregiving patterns, breastfeeding intensity, maternal-infant separation, maternal-infant dyad sleeping pattern, co-sleeping, and the richness of the allomaternal network. And so if you remember, those are the 4 ancestral caregiving traits that I was talking about earlier. And so trying to tie this together, whether or not those caregiving patterns are related to variation in the HMM. And so using linear mixed effects models, we found, I did not bother to put up the table because that's almost impossible to read and nobody wants to look at that. But we found that many of these features are associated with HMM richness and Shannon diversity, explaining 22% of the variation in HMM richness when controlling for geographic variation. So we found, for example, that parity time postpartum in animals increases the milk microbiome richness, while age, BMI actually is only a trend, household members, and the amount of time mothers and infants are separated from each other during the day decreases HMM richness in moms. In terms of diversity, we found that these factors explain about 13% of the variance, and we found that parity, time postpartum, co-sleeping just a trend, and the richness of the allomaternal network increased diversity in mother's milk. So just to point out, kind of supporting what we found in the CAR, while age, breastfeeding intensity, and the length of time moms and infants were separated from each other during the day decreased the diversity of the community in mother's milk. In terms of whether these features were associated with the specific taxa in milk, we took a conservative approach. I'll just go really briefly over this instead of getting too much into the weeds. But we conducted an analysis of composition of microbes, or ANCOM, and ANCOM identified multiple taxa that are significantly associated with variation in caregiver behavior. And so I'm just focused on those 4 ancestral caregiving traits right now. But after we conducted ANCOM and then identified the significant taxa, we then conducted a follow-up regression to look specifically at the ones that had a significant linear relationship. And then we also did visual inspections of diagnostic plots to further reduce potential noise to make sure that these relationships weren't being driven by one population or 2 populations. And so what we found is, for example, that there is a significant negative relationship between diela and breastfeeding frequency. Mothers who breastfeed more frequently have significantly lower relative abundance of diela in their milk. We also found that Xanthanobacterium, also identified as an important taxa, both of these were, in the Central African Republic, was significantly and positively associated with the richness of the infant's allomaternal network. Mothers of infants with richer allomaternal networks have a higher relative abundance of Xanthanobacteria in their milk. Now, all of these analyses were done at the genus level, and so it's not possible to discuss functionality at the genus level. Species within the same genus can be commensal or pathogenic or both. Nevertheless, we can group these into different categories. And so if we look at the ones that are highlighted, these are generally related to aquatic or soil environments. And the negative correlation between breastfeeding intensity and such microbes may reflect the fact that as breastfeeding and contact increases, there is less opportunity for environmental microorganisms to colonize the baby and or the breast. This would help explain why breastfeeding intensity and co-sleeping, closer, longer, and more intense contact with the mother, are negatively associated with soil and water-related bacteria, while allomaternal networks are all positively associated with those bacteria. Perhaps we're seeing that because there is a reduced or less intense contact between the infant and his mother or her mother when he or she has more allomothers. And so all of this opens up more questions than we are able to address yet, but it is a really exciting time to consider how human caregiving behavior and how our social worlds, integral parts of the evolution of human development, are associated with our microbiomes. We are just, really at the tip of this. We are expanding to other milk components. Milk contains hundreds to thousands of bioactive factors, and increasing evidence suggests that milk is highly dynamic and responsive to not just the physical environment, but also the social environment in which the mother and infant dyad reside, which, given our history, our evolutionary history of social and caregiving patterns, makes a lot of sense. Anthropologists have for decades recognized the importance of human sociality and social networks on infant health, development, and well-being but the pathways of how allomothers were influencing them have been elusive. And so perhaps this interplay between our social environments and breastfeeding and milk composition offers one potential avenue of influence in how they might be improving or degrading infant health. Our results suggest that there may be multiple pathways via which the HMM is shaped, but one particular pathway of interest may be that via the infant oral cavity. We posit that the infant may be signaling their environmental exposures broadly construed to the mother during breastfeeding. Our future studies need to explore infant growth, development. We need more longitudinal studies that look at health outcomes associated with HMM and the maternal-infant dyads environments. And as we, and I mean the global we, move forward in these studies, I hope that I can encourage others to engage in this anthropology of microbes. We are just beginning to understand the implications of human-host interactions, and bringing an anthropological perspective is essential. We have a fundamental understanding of why cross-cultural and non-Western studies are essential. We also frequently bring a theoretical perspective to the table. And so what I've talked to you today is limited to 16S data, but since beginning these studies, the research and the methods have just expanded dramatically and are moving into multi-omics. And so in the future, we should be able to get a better hand on functionality. And so it's just been a great and exciting time to be a part of this work. And so I'd like to thank all the mothers and infants who participated in the NSF Career, NSF Inspire, and HERC projects, and my gratitude to all of our local assistants in the CAR, Ethiopia, and all of the local teams who participated in the Inspire project. And with that in mind, I really do need to highlight the amazing team that is the Inspire project. I believe in interdisciplinary research, and when we started this project, we reached out to the world's experts in immunology, bioinformatics, microbiology. We have anthropologists, lactation physiologists, nutritionists, you name it, they're on this team. And we have just an amazing team. And I am incredibly grateful to all of them for their contributions and collaboration on this project. And so, thank you.
When the allo mothers are with the infants just up to 6 months, are they nursing the infant?
It depends on, in the Central African Republic. Frequently, yes, not always. It really depends on where the mother is residing. Is it her family? Is it her sister? And so it varies, but we definitely see it in the Central African Republic. In the Inspire study, we see quite a bit of it in Peru as well.
And then are any of these cultures using supermarket milk?
So not those that are, well, let me start that sentence over again. Not in the Central African Republic. In the Inspire study, moms did not have to be exclusively breastfeeding to enroll, and throughout the world, many moms are giving their infants water or other semi-solid foods and liquids, but we didn't have, these infants were breastfed, not formula fed.
So lots of sources of variation.
Yes.
Potentially. And is your study going to be looking at the infant microbiome?
Yeah, so we have the infant fecal microbiome data. We have 2 papers out on that right now. One, it was part of the first paper on the microbiome that Kimberly Lackey published last month, and my graduate student, Avery Lane, just published a paper last month as well, looking at how household composition influences the infant fecal microbiome.
It's just an amazing area of research. There's going to be so much more coming out of it. You know, we have more microbiome cells in us than we have our own cells, you know. And I'm really happy to see that you're starting to explore all these things.
Yeah, it really is an exciting time, and we've just been having a lot of fun doing it. So it's been a lot of work, but we're having a lot of fun.
Thanks for a fascinating talk. Do the infants in these studies consume soil? Do they eat soil?
Well, these are all fairly young infants, so is there a possibility that they have had soil enter their mouths? Probably. I mean, I would think that would happen in the U.S. too.
Yeah.
And so, yes, I think that they probably have consumed soil in terms of just environmental contact. But they're fairly young too, so they're all, well, I shouldn't say that. For CAR, that was a much greater range. Yeah, but in the INSPIRE study, they're all still little. So they're not having quite as much environmental exposure. It's unlikely that you're putting a 6-month-old, you know, or I should say a 2-month-old on the ground. Yeah. So.
Okay, sort of a related, really technical question. So why clean the breast before taking the milk sample? This is something I asked you too, because so like Himba moms, for example, where they put ochre on their breasts, and it's clear that the babies are ingesting that when they're breastfeeding, and so is it like it seems like a sort of methodological choice, I guess, to decide that you don't want whatever they would normally be consuming that way.
It's a really good question because if you want to know what the baby is getting, they're getting everything that's on the breast. And so it really, I guess it depends on the question that we're asking. And in part, some of this is because in the early stages of this research, and not just ours, but in general, there was an assumption that it was contamination. And we've looked at that. We actually are looking at that more now, and doesn't look to be contamination. But so in that respect, that's part of the reason that we did it. And then in the international study, we really needed to standardize as much as possible. But on a personal level, I feel like if the infant is taking it in, then that's what they're getting. But that would relate more to what's getting into the infant versus what's in the milk. Yeah.
Thank you so much for this great talk. I was wondering about methodologically also time of day as difference, and the second question was, you had said there's no normal milk composition, and then I remember you were talking about the HMM as having novel things, and so I was wondering if you could talk about the variation of milk composition as how much of that is novel and how much of that is variation of different levels of things.
Sure. Well, in terms of the variation across the day, we did standardize that. I didn't mention it, but we collected all the samples in the morning. Moms should have nursed from their breast in the INSPIRE study for, I have to go back and remember, but we did standardize that. And then in terms of the no normal, we see that, I guess maybe I could use a little bit more clarification from you, sorry.
Oh, I was just kind of wondering how much of the difference among populations was that there's novel things in the milk, so like an immune factor that is in this population but not this, versus just the variation was extreme of different. It had the same stuff but different levels, or how much was just difference?
Oh, so, well, it depends on what factor we're talking about, but I would say both of those. We do see some, novel taxa, and we do see that, there's just variation in the relative abundance, let's say, across populations. So even if we're talking about some of the more basic, I don't have the slide with me, but even if we're talking about some of the more basic components like lactose or protein, we've always assumed there's a fairly stable level or a normal level. And when we've looked at it from the INSPIRE milk samples, some populations are significantly different from that. And so what we thought of as standard might not be standard. And it's not that one population has it and another doesn't. It's simply that the amount is different.
Thank you.
I'm not sure I answered your question,
No,
did
yeah.
I?
Yes, thank you.
Okay, I'll try again if I didn't. Okay. Yeah.
So, the effects of co-sleeping on the HMM, does that boil down to just frequency? That there's, because I can't see any other pathway to that. I mean, frequency makes sense overall in terms of, as you said, decreased opportunity for colonization. But beyond that, I can't see why co-sleeping should matter.
So I think a couple of things there. One, our frequency or intensity of breastfeeding did control, it was based on both day and nighttime breastfeeding from self-reports. That was not behavioral data, but self-reports. So that should technically be controlled for in the model. And so in part, I think it might be exposures, different exposures in the bed. We're still trying to figure it out, to be honest. And it might be related to maybe not necessarily the frequency, but those number of bouts, and that's why it's coming out like that. And honestly, we don't know yet, and so we're trying to tease it apart still.
And forgive me for, there's so much information. I mean, it's really impressive how much you've collected, but did you have data on HMM and co-sleeping in Sweden as well, or is that a different study?
Oh, yes, so Sweden was in there. The one population that was not in those data was the US California population, because we didn't get full survey data on that population.
The reason I ask is because if you think about, say, environmental variation between the sleeping environment and the rest of the environment that the mother and infant occupy, with regard to just, say, a variety of bacteria present, you wouldn't expect there to be that much difference. But you might expect there to be difference with regard to, for example, ectoparasites. Presumably those are absent in Sweden, so it seems like Sweden is a critical case for trying to pin down what the mediating variables are and the effects of co-sleeping.
We'll have to kind of start to tease them apart now. I mean, it's a really good point, and we can look at some of, we have some populations where we're able to dive into some of these questions more deeply. And so we haven't done it yet, but it's a really great point, and we can try to dive into those. Yeah.
This is a little bit more of a basic question, but something that I find really interesting with Dr. Schultz's work as well. But how were you able to, especially with such a large cross-cultural sample, what was your process of like talking women through giving up their breast milk? Like as far as like the preciousness of what they need for their baby, and also as far as like personal space and those sorts of things?
Well, it's a great question. And one, we collected, we try not to take more than we need, and for the analyses, and we had several different analyses on here. So, but even so, this is in part due to this group of people up here. When we started this project, we reached out to people who we've known and who have engaged in this research around the world and have well-established relationships with their communities that they work in. To all of us, that was really important to make sure that we're doing ethical research, make sure that moms are aware and engaged in what we're doing and understand why we're doing it. And so in part, that comes down to working with amazing people and working with people who have long-term field sites. And so we're not dropping in and saying, hey, can I have some milk, and then leaving. Yeah.
How much did you wind up collecting?
We collected one, I'm trying to remember the different studies here. We had 1 ounce on the Inspire.
Okay.
Oh, for the I'm sorry, for the microbiome, I should say. I have to go back and look, honestly. Yeah.
About how many people were in each study from the different areas?
So we have small population sizes on those. We have about 40 women per site on that. Yeah.
Is there microbial composition differences in formilk, hind milk, anything like that?
Oh, there's so much left to be done. And so, we're It's on a list. I think it's on many people's lists right now to start to be able to tease that apart.
So I'm just thinking about the cooperative breeding and the household composition kind of data that you have access to. And so unlike a sort of US or even probably a Swedish nuclear family kind of style where you would have, unless you had twins, right, you have one baby in the household, right? And so you've got maybe still an allo-maternal network, but with that baby being the sort of central node of it, right? Whereas in other kinds of households, you would have multiple babies, right? And the babies are maybe more or less interacting with each other, depending on how the household is set up. And then, like, if each one of those babies is sort of a central node to their caregiving network, and then the babies are interacting with each other and presumably touching each other in all kinds of ways where they could share things, it seems like that would set up, I don't know, I'm just wondering whether there's some sort of like interesting way to look at those networks and see whether something about the number of babies in the household actually is a factor that matters too.
I would imagine it might, I don't know, but I would imagine it might become more important as the infants move into later infancy than these early couple of months. But I certainly think that those networks are really important. And that's why when looking at the study in CAR or even the INSPIRE study, and that variation or the diversity in the breadth of people engaging with the infant, I think that's why we're seeing that it's important to the microbial community. Because if you only had one type of caregiver engaging, and that caregiver only went to one environment, let's say went to the field every day and came back, you would only see that influence. But here, because we can document that there's real diversity and breadth in terms of who's engaging with those infants, all of those environments that all those people are going out to are coming back toward that infant and the mother.
Why did you decide birth to 6 months? And do you think that, because lactation usually continues longer than that, and do you think that the composition of the milk changes as the infant matures?
So for the INSPIRE study, it was 1 to 3 months. We had a couple infants that were outside, so it was really 1 to 5. But for that was to keep it consistent or to make sure we had a tight enough window that we could compare. For the CAR study, we had a greater range in age of infants nursing, which is not surprising among a hunter-gatherer population. In terms of milk composition changing over time, yes, milk composition does change. It depends on what factor we're talking about. And so we see in the INSPIRE project here that we do see some effect of time postpartum on the microbial communities. But even my colleague Shelley McGuire and Mark McGuire, their work with a longitudinal sample did not show much variation. And so that question is really up in the air still. Did I get all of those parts? Okay.
Great talk. Really amazing field that you're helping develop. This is wonderful stuff. And I'm thinking about the communication between the infants and mothers, and wondering about there's the idea that mothers are priming their young ones to be able to be, confident, especially with the immune system and the environment that they're going to be, more and more having to handle, independently. Is there also a side of the story about how there's not just a process of priming, but also filtration of mothers, given a life history of like an optimal timing of when to introduce immune factors to the offspring? Like, are there some immune factors that are present and that are useful for an adult, perhaps, but are something that you'd expect not to be, adaptive to be introducing to a 6-month-year-old necessarily? Or is there a model there for the kinds of filtration and titration of immune factors to children?
I I wish I knew the answer. I'm definitely not an immunologist, and so I don't know. I actually have one of my graduate students who's is going to start to look at some of the immune factors in the Inspire milk, but with some help from people who are better with that.
Yeah.
But I honestly, I don't know the answer.
Do you, so like I work with the Hadza, you know, and so adults are probably carrying around with them, you know, acquired immunity to things like bee stings, which would be great to have for a 10-year-old. Will you have data that allows you to compare the profile of the adult acquired immunity to the very young, to be able to track like the life history of the development of acquired immunity, or are those data, like, do you have paired data with adults as well as offspring, young infants, to be able to compare the immune factors that they each have? Or?
Yeah, so we have immune specificity data on the mother's milk, and we also have when we have infant fecal samples.
Okay.
But we haven't done the type of analysis that you're talking about yet.
Okay, so like blood samples from the moms, you don't have?
No.
Okay.
Thanks. I'm wondering if, and especially in the wealthy countries, the US and Sweden, if there's variation between moms or between households in, I guess what you call sort of antimicrobial behavior, like with some moms, being very concerned with washing their hands before they touch the baby, and then when the baby starts crawling to like make sure the floor is clean, and other moms being more casual about it. Any hint that something like that's happening?
So we don't have that. We don't have that data in the Inspire study. We have. We could sort of get at it, but nothing specific. We've also just completed data collection on another U.S. project where we looked at things like how often they were washing that pacifier if it hit the ground and things like that. But we haven't analyzed those. We're just analyzing the samples right now. So.
Thanks.
Yeah.
Just kind of following up on both Joe and Brian's points. So I understand you don't. Not only do you not have the results of those analyses, but it's not clear that you even have at present access to the kind of longitudinal data that would be necessary to test Brian's postulate. But it's important to keep in mind that suppose that you got a result that looked like confirmation of Brian's hypothesis. So suppose that moms have antibodies to X, right? Infants start off, and there are antibodies to X in breast milk. Okay? Infants start off without antibodies to X, and that at some time down the road, potentially post-weaning, offspring display antibodies to X. Okay? The maternal, as I understand it, the maternal antibody transfer is temporary. That is, those are not actually colonizing the offspring, and potentially what those provide is an umbrella during which the offspring can get exposed to those antigens without suffering pathogenic effects, and subsequently, as their own immune system matures, develop acquired immunity to those pathogens. So what looks like inheritance actually isn't inheritance at all. It's just more like scaffolding that provides an opportunity for learning, right? So the design of those kinds of longitudinal studies has to entertain those two possibilities. And in fact, Betsy Abrams and I have a paper in which we argue that is precisely the function of infant mouthing behavior. The reason that infants put everything in their mouths, including dirt from the floor in the variety of circumstances, is to expose themselves to antigens while they are under the sheltering umbrella of the maternal antibodies. So, as you think about future investigations, I would encourage you to think about the richness of the possibility space.
Yeah, thank you.
Other questions?
I have a question, since that wasn't really fairly described as a question, it was more a commentary. So I saw that you had parity on the list. Do you have any ideas about how and why parity might matter?
Well, for the HMM, and because, so there was that transfer of the milk was shown by Ramsey in an ultrasound. And so milk is basically pushed
Right,
back
backwards.
into the breast. And so that's why I think that parity would be a factor. Because your siblings, your previous siblings, might be influencing your mom's milk.
Right, so can you test that across your study sites by looking at the frequency of allomaternal nursing, right? Because if you're an only child, but mom is nursing lots of other people's babies, then it's as if you had many siblings in that context. Right?
So we had, overall, we have a couple sites where it happens. The numbers are fairly low. Peru had a lot of allo nursing, and we have data on both whether mom ever nursed another baby or whether the baby's nursed by others, both of which could be influential. And we haven't looked at that yet. So we have mounds of data right now that we just need to kind of plow through. But yeah, so we haven't done it yet. But I agree with your point. Yeah.
More questions?
I have one quick one. With this amazing data set that you collected across all these societies, will you have follow-up to track, for example, mortality of the infants through time?
No, anything that we do longitudinally, we're so this was cross-sectional one time with these moms, and so, but anything that we do longitudinally will come from a new study.
Okay.
Yeah.
Okay, great, thanks.